be here. I'm from Queen's University. I actually work in Dr. Suchek's lab. And I actually really like this picture of campus because it's the only time that it's not snowing in Kingston. But today I'll be talking about the role of the GPCR receptor, G-protein coupled receptor, and how it activates insulin receptor glycosylation. So the mechanism of insulin action is actually very well characterized in the literature. Once insulin binds to its dimerized receptor, it can initiate cell signaling through the activation of insulin receptor substrate 1, which becomes phosphorylated. So phosphorylated IRS1 can lead to the activation of two signaling pathways, which can result in cell proliferation, differentiation, and survival. This pathway, kind of known as the RAS-MAPK pathway, plays a very prevalent role in cancer, but a metabolic role in the cell through the PI3K-AKT pathway to upregulate the presence of glucose transporters in the cell, increased intracellular glucose concentration, as well as the production of glycogen and lipids in the cell. However, if any of these components are abnormal or insulin fails to bind to its receptor or the receptor malfunctions in terms of its activation, it can cause a wide range of clinical disease, such as insulin resistance, diabetes, cancer, and so on. In terms of its glycosylation, the insulin receptor is heavily glycosylated. It has about 18 N-glycosylation sites, 14 that are confirmed. And in our lab, we look at the glycosylation in the beta subunit. So the insulin receptor has two extracellular alpha subunits, which bind hormone ligand, and two beta subunits that are transmembrane and cytoplasmic. And the cytoplasmic domains become autophosphorylated when the receptor is active and initiate cell signaling. So IR glycosylation has a very important role in terms of the function of the receptor, in terms of its processing, transport to the cell membrane, recycling, even its ability to bind hormone. However, we're pretty much focusing on the role of glycosylation in terms of autophosphorylation of the beta subunit, so the N-glycosylation sites in the beta subunit, and how glycosylation impacts their ability to associate, autophosphorylate, and so on. Specifically, we're looking at that terminal sialic acid residue that Dr. Suchek mentioned. The enzyme that cleaves that terminal alpha-2,3 sialic acid residue that we are focusing on is the neuraminidase 1 enzyme. We found that this enzyme plays a really important role in removing steric hindrance from that receptor and activating the receptor. So when we do immunofluorescence protocols, we see that when we add insulin to cells, we can detect a high presence of phosphorylated IR beta. So phosphorylated IR beta is our indication that the insulin receptor is active, and that's quite significant compared to the no ligand control. However, when we add specific neuraminidase 1 inhibitor, Tamiflu, in which the free acid is structurally similar to sialic acid, so it competes for the active site, we see an inhibition of the ability of the receptor to autophosphorylate even in the presence of insulin. We add PPP to these cells in order to inhibit the insulin growth factor receptor, because insulin can bind to both IGFR and IR, so we just want to control for that. In terms of the signaling paradigm that we kind of use to understand how NEU1 regulates these inhibitory processes, our lab has kind of proposed this signaling axis where when ligand binds to its receptor tyrosine kinase, so in this case, insulin binds to the insulin receptor, you get subsequent activation of a GPCR complex that our lab's identified to be the neuramin B GPCR. When you activate GPCR, you induce G-alpha-I signaling, which activates matrix metalloprotease 9. MMP9 activity has specific elastase activity to remove the elastin binding protein that forms a complex with neuraminidase 1 as well as protective protein cathepsin A. Removal of that EBP from the complex is what allows NEU1 to be active and catalytically cleave that alpha-2,3 sialic acid residue. Again, this is the beta subunit of the IR receptor, and removal of that sialic acid residue allows the beta subunit to associate, autophosphorylate, and elicit cell signaling. We perform sialidase assays in our lab that involve adding substrate to live cells and measuring the production of fluorescent product. So the more active neuraminidase 1 is, the greater fluorescent product it produces, and that's a bright blue color. We notice that insulin, at its highest concentration, can induce a significant amount of sialidase activity in a dose-dependent manner. So when you decrease insulin concentration, you don't see as potent of a sialidase activity. To show that it's NEU1 specifically and not the other neuraminidases that are playing a role in this regulation process, there's actually 4 malloneuraminases, and we wanted to show that it was NEU1. We added neutralizing antibodies individually to these cells to show that only when you inhibit NEU1, you see that decreased sialidase activity, and that doesn't happen when you inhibit the other 3 neuraminidases. Again, since Tamiflu is a neuraminidase inhibitor, the same pattern can be seen here, also dose-dependently. So the highest concentration of Tamiflu dose-dependently inhibits sialidase activity. So we also wanted to show the role of those other protein complexes in terms of sialidase activity. So galardin is a broad-range matrix metalloprotease inhibitor. Again, dose-dependently, we show that at its highest concentration, galardin inhibits that fluorescence. And again, analyses were done to show that MMP9 is the responsible matrix metalloprotease for this activity, also in a dose-dependent manner. Also, to kind of further confirm that signaling axis, we wanted to look at the role of that neuraminin B GPCR. As Dr. Suchak mentioned, bombazin is a specific agonist for that GPCR complex, and addition to bombazin alone is sufficient to induce sialidase activity. We can inhibit bombazin-induced sialidase activity using a specific neuraminin B GPCR inhibitor called BIM23 to see that there's a partial reversal when you add both. As I mentioned, this is the proposed signaling axis, and this is our lab's model in terms of how ligand binding induces neuraminidase 1 activity. And we wanted to show that these components are localized together on the cell surface, and we wanted to show that they don't come together upon ligand binding. They actually exist without the need of ligand stimulation. So we can see that if you precipitate NEU1, you can also pull down IR beta, and that happens when cells are stimulated with insulin, and if they're not. So they don't need insulin stimulation in order to become associated. The same can be shown with MMP9 and IR beta, as well as the neuraminin B GPCR and IR beta. So this kind of tells us that, yes, these complexes are associated on the cell surface. They don't need ligand binding in order to associate. So then, what can we do with this? Well, in instances of insulin resistance or type 2 diabetes, for example, even sufficient production of insulin isn't sufficient to induce cell signaling in terms of the metabolic role of insulin. So maybe we can target the activation of the insulin receptor in the absence of insulin using some of these other molecules. And my specific work involves the activation of GPCR using different types of agonists, and perhaps maybe that activation can indirectly activate the IR in the absence of insulin. So this is that same sialidase activity, but using GPCR agonists. We use insulin as our positive control, and we can show that bradykinin, angiotensin 1 and 2, in the absence of insulin, can induce sialidase activity, but when we add that specific GPCR, neuraminin B GPCR inhibitor, the BIM23, we can reverse that process. Okay, so GPCR agonists are sufficient to induce sialidase activity, but what about to induce IR activation? Well, as I mentioned, we look for evidence of phosphorylated IR beta subunit to tell us that the insulin receptor is active, and we can show that when we add these agonists, we get IR beta phosphorylation when we do immunofluorescence protocols. We also wanted to show that other downstream proteins of that insulin signaling axis can also become phosphorylated. So we also looked for the presence of phosphorylated IRS1 or insulin receptor substrate 1. Again, similar results were deduced. Insulin, again, being the positive control, we used 4 GPCR agonists to show that GPCR agonists alone and without insulin can cause phosphorylation of IRS1. So in conclusion, my talk's not as long as the others, but we showed that you don't really need insulin in this case. If you can stimulate the GPCR, in this case the neuromin B GPCR, you can elicit the activation of these molecules to stimulate NEU1, to cleave that terminal alpha-2,3-sialic acid residue, and induce the cellular effects of insulin without the requirement of insulin. And I'd like to thank my supervisors and current and past lab members, and thank you.
Can we have questions for the speaker, please?
Only none from Dr. Suchak.
Other questions?
If you use the base, then
Sure.
you don't have to use the insulin.
In terms of phosphorylating the insulin receptor,
Yeah.
yes, you don't need to, but this is all in vitro work, so I haven't studied the toxicity of bombazine in mice or anything like that. This is just in terms of characterizing a signaling pathway.
Question is that when you use anti-NEU1, actually you see the decrease of phosphorylation. So my question is that the NEU1, do you really, when you do cell culture work, do you really see that in secretion in the media, or is really associated with the membrane itself? In your drawing is like buried.
Are you asking if NEU1 enzyme is in the cell membrane?
Or like in the cell culture, do you actually find in the sugar in the cell culture?
Well, there's 4 neuraminases. Some of them are cytoplasmic, but then NEU1 and NEU3 are on the cell membrane. There are some that are also in the cytoplasm. Does that answer your question?
If it's in membrane, how the antibody can work that way? So it must be, outside the cells.
Yes. I mean, we see evidence of NEU1 coming into the cytoplasm, but that's just in terms of it being recycled. So once it's active, we see that after 5, 10, 15 minutes. Sometimes it becomes internalized and then it comes back out. But in terms of NEU1, it's not a cytoplasmic enzyme. That's right.